All right, everybody. So today we're trying something a little bit new. As you can see, our speaker is not physically with us. Dr. Nancy Gagnon is actually in her office at MIT, and she requested that she give this lecture remotely as part of our effort as academics to reduce greenhouse emissions, and hopefully that can take over a little bit more. And we can, be more environmentally conscious as a profession. So Dr. Kammerscher did her bachelor's and PhD at MIT. Then she went on to have active positions at UCLA and then Harvard before returning to MIT in 1997. There she has done a lot of pioneering research with fMRI. And she has especially focused on this idea that there's modular structure to the brain, and she has explored that really for the next several decades. So we're very lucky to have Nancy talk to us today. And without further ado, Nancy. Oh, and by the way, if you have questions, we need to ask on the mic. So if there's a clarifying question, just shout it, and I'll repeat it in the mic for Nancy. And questions in the end, I'm going to ask you to come here so that Nancy can see you as you're asking the question. Okay?
Great. Thank you so much, Toby, and thanks to all of you for humoring me in this kind of experiment on a virtual visit. I know it's not exactly the same as being there, and there's some risk of technical snags, at least at my end, if not at yours. But I think, as Toby said, the potential for reducing carbon emissions is something all of us academics need to be thinking about. So, can I just first ask and make sure, you can hear me okay? Yeah? All right, awesome, great. Okay, so in this talk, I want to start with some good news, which is a broad overview of what I think is really some substantial progress we've made in our field over the last 30 years in understanding the human brain. And then I'll go on and talk about 3 questions in kind of ongoing research that try to build on that progress to take the next steps. So let's start with the overview. To appreciate how much progress we've made, I think it helps to step back a few decades to what we knew about 1990. And that's shown schematically here. From studies of patients with focal brain damage, we knew about the very approximate location of the brain regions engaged in perceiving faces on the bottom of the right hemisphere, language, the lateral sides of the left hemisphere, and attending to different objects up in the parietal lobes. But that was kind of it. And importantly, that could have been it. Back in 1990, it just wasn't obvious that if we had better methods, that we would be able to see a much finer grained organization of the brain, because maybe there wasn't any finer grained division of labor with different processes in different locations. But then functional MRI came along, and here's where we are today. There are now dozens of regions of the cortex for which we have a pretty good idea of what that patch of cortex does. And each of these regions shown here is present in pretty much the same location in more or less every normal person. Now, this is a highly schematic diagram. We don't have neat little colored ovals in the brain. So let's look at some real data to show what I mean by this diagram. So here is a brain image of one of my fabulous lab techs, who's not only a great tech, but a good sport, getting scanned a lot. And what you see here is the bottom surface of the brain. Can you guys see the pointer down here when I move the pointer around? Yes, great, okay. So this is the right hemisphere on the right, left hemisphere on the left. We're looking up from the bottom surface of the brain, frontal lobe, occipital lobe. This is the bottom of the temporal lobe here. Okay, and this picture shows the cortical surface mathematically inflated so you can see the whole surface. So the dark bits are the parts of the cortex that used to be inside a sulcus until they got mathematically inflated to show the whole surface. In color are the functional activations. And what that means is, for example, this little blue patch right here on the bottom of the right fusiform gyrus, here's its response when this subject is looking at faces. Actually, this is pooled data from many subjects. the analogous region. This is a response when they look at faces, bodies, scenes, objects, words, and other things. So you can see the response is substantially higher to faces than to any other stimulus class. Right next door, this little green patch responds very strongly when people look at scenes and much less strongly when they look at all these other classes of stimuli. Over here in the left fusiform gyrus is a very special region, sometimes called the visual word form area, because it responds very strongly to visually presented words and letter strings, much more strongly than to any other class of stimuli. And out on the lateral surface of the brain up here, there's a little purple patch right in there that responds selectively to bodies. Here's a response to images of bodies, hands, feet, legs, nothing interesting, and much lower response to all the other different stimulus classes down there. Now you might think, looking at this, that, oh, sorry, forgot one. Over here on the left temporal lobe, there's a region that responds selectively to speech sounds. So this is response in dark orange to the sounds of a language you know, and in light orange, the sounds of a language you don't know and don't understand. So this orange region is a response to speech sounds, not the meaning of language. So all of these cases are cases of high-level perception, where we're discriminating one class of visual stimuli. So you might think, well, maybe we have special brain machinery only for specific kinds of perceptual discriminations. After all, David Marr reasoned way back in 1980 and before that aspects of vision, at least, were very compartmentalized, and the sets of computations that you conduct, for example, when you perceive visual motion, are really quite different than the computations you have to perform to perceive color or shape or recognize objects. So is this something that's found only for perception, or do we have brain specializations even for higher level mental functions? Working with Ev Fedorenko, starting a few years ago, Ev was then a postdoc working with me. She's now a faculty member in my department. And Ev first figured out how to identify language regions in the brain. That's this set of red regions here in the left temporal and frontal lobe. And as I showed you on my first slide, it's not a surprise to know where those regions are. What Ev did was develop methods to identify them very precisely in each subject individually. And that's important because although I said every subject has these regions in the same approximate location, their exact anatomical location varies a bit from subject to subject. And so if you want to study them, you have to identify their exact location in each subject individually. Then having done that, Ev went on to ask the question of whether those regions are really selective for language per se, or whether they're engaged in a whole host of other mental functions. And this has been a long-running, fraught question for essentially hundreds of years in our field, whether, from Broca and Wernicke to Chomsky and everyone between and since. And so Ev decided to test that question by identifying those regions in each subject and then measuring the response in a whole bunch of other tasks. And I know that this is too much information and you can't read it, but it doesn't matter. Point is, there are lots of other tasks we tested, arithmetic, music, working memory of various sorts, all the mental functions that people had hypothesized overlap with language in the brain. And what Ev found can be shown here. So this is basically Wernicke's area, language region in the temporal lobe. Here in the black bar is its response when you read sentences. Here's when you read a string of non-words that have no meaning. And here is a response in all those other tasks. Basically nothing. So what that shows is that this region is extremely selectively responsive when you understand the meaning of the sentence, and it's not responsive to all these other tasks. Lots of other brain regions are responsive to these other tasks, in fact, often very nearby regions, but not the language regions per se. You see the same deal up in what's basically Broca's area, up here in the frontal lobe, strong response to sentences, much lower to unintelligible non-word strings, and little to no response in any other task. And I really love this result because to me, it's not just another specialized blob on the brain, much as I do love those, but this is a very special blob, and I think it's telling us that language is not the same thing as thought. And probably every one of you has wondered at some point in your life what the relationship is between thought and language. Philosophers have been writing about this for a very long time. Do we think in language? Could we think if we didn't have language? And I think this is a beginning of an answer to that question. In the brain, thought and language are really very non-overlapping. They're not the same thing. And by the way, even stronger evidence for that conclusion comes from work, not in my lab, but from Rosemary Varley in England, who has been for a very long time testing a set of patients who have global aphasia due to massive left hemisphere strokes. And this is a very awful condition in which you basically lose all language, not just your ability to produce language or just to understand language, but you lose all of this. You basically don't have language at all. But she's very cleverly figured out how to communicate with these people and to test their ability to do all these other mental tasks that you might have thought require language. And what she finds is that these people can do every one of those tasks, even though they lack language. They can do arithmetic, they can appreciate music, they can do logic problems, they can think about what another person is thinking. And so that shows you even more strongly that once you've lost language, you can still think in many different kinds of ways, and language and thought are not the same thing. Okay, moving along, perhaps the most astonishing finding in this whole line of work is the work of Rebecca Sachs, shown here. And she studied for a long time this little pink region here, known as the right TPJ, because it's in the temporoparietal junction. And what Rebecca showed is that region responds strongly when you think about what another person is thinking, and not when you think about a whole bunch of other kinds of things. And what's remarkable about Rebecca's work is that she's shown how specific this region is by testing it again and again and again on a whole set of things to really kind of hone in on what exactly it does. And remarkably, this region doesn't respond when you think about another person's appearance. It doesn't respond even when you think about bodily sensations like thirst and hunger and pain. It only responds when you think about the contents of their thoughts. And if that strikes you as crazy, reflect for a moment on the fact that we do this all the time. Being a human being is basically spending a large percent of your waking moments thinking about what other people are thinking. It is the essence of art and literature and humanity to be thinking about what other people are thinking. And so perhaps it's not that surprising that we have a specialized bit of brain for just that mental function. Okay, so lest you guys think I'm completely nuts, I want to emphasize that I don't think that every patch of cortex is specialized in this fashion. In fact, these white regions up here are indicated in white and not with a color to indicate that in fact they're kind of like the opposite of this specialization of all these other regions. These white regions have been called the multiple demand regions by John Duncan because they respond to multiple different kinds of cognitive demand. Virtually independent of what the task is, if you're doing something difficult, those regions turn on. So here are a bunch of different tasks. The details don't matter except that the filled-in bars are the difficult version of the task, and the light bars are the easy version of the task. And you see across a wide range of tasks, they respond more to the difficult than easy condition, and they don't care very much which task it is. So that shows you that in addition to these highly specialized cortical regions, we have some very general purpose regions, which actually I think in the long run are more computationally mysterious than the specialized ones. I mean, what are these? The brain's CPU? I don't know what they are, and I think it's a fascinating mystery. Okay, so that's my whirlwind overview of some of the progress that the field has made over the last couple of decades. Just to summarize what I think is cool about this, I think this is real progress. I think we found a set of functionally distinct regions of the brain that are present in pretty much everyone. And these include regions specialized for abstract, uniquely human functions, like understanding language, appreciating music, or at least perceiving music, and thinking about each other's thoughts. And I like to think of this diagram as kind of an initial sketch of who we are as human beings, as thinkers. This is our set of basic machinery that we all share. But you can also see that this is just the barest beginning. It's very rudimentary to just say, okay, here's a patch of brain that does face recognition. Well, what does that mean, right? It just leaves open a whole host of questions. So really, I think the way to think about this diagram is as a roadmap for future research. one that opens up a vast landscape of questions. So today what I'm going to do is address 3 of these questions, starting with one that's probably already in your head, and that is, really? Some regions just do one thing? I'm going to talk about that a bit, but I want to heighten the importance of this question by bringing up a line of work that some of you are probably familiar with, but others may not be, and that is the results from what's sometimes called multiple voxel pattern analysis. So to review that very briefly, way back in 2001, Jim Haxby published this very important paper in which he showed that if you look at the pattern of response across the whole ventral visual pathway, like this whole region back down in here, you can pull out all kinds of information. And most relevant for present purposes, if you looked at just that fusiform face area, just looking at the pattern of response in that region, you could tell if a person was looking at chairs or shoes. And that's a very important challenge to the story that I'm pitching here, that region is very specialized just for face recognition. After all, we care not just about activation in the brain, we care even more about information. And if that region holds information about chairs and shoes, what am I doing saying it's specialized for face recognition? So that's the challenge raised by pattern analysis, and I think it's actually the most important, substantive, serious, and interesting challenge to the functional specificity view. And so the essence of the idea is, if face areas contain information about non-faces, that's what I just said, that's an important challenge. But here's a possible response to that challenge. Wouldn't this be expected of any dedicated face processing system, even one that had been designed all and only for face recognition? After all, if you take a shoe and you project it into face space, you probably get a different representation than if you take a car or a chair and project it into face space. And if so, then even finding information about those non-face categories wouldn't be evidence against the hypothesis that the fusiform face area is optimized all and only for face perception. So that's the possibility, but how could we test it? Well, here's the idea that we had recently, and that is to use a deep neural network trained only on face identification as a proxy for a face recognition system, and ask whether it has that property. So in particular, this is work with my postdoc Katarina Dobbs. So what she did was to take a variant of AlexNet, train it on 1,000 faces from VGG face, with many different images of each face, train up a face classifier with all 1,000 identities. This is our proxy for an optimized face recognition system. After all, this network has only ever seen faces, and it's only been trained on face recognition. So you can think of it as a kind of ideal optimized face system. And indeed, after training, it's very good at identifying novel images of trained faces. Much like many of the results in the literature, these things are now near 100% correct at this task that nobody could even imagine how to do a mere decade ago. But now the key question is, how would this network do at discriminating objects? Would it manage above chance, as Haxby showed for the fusiform face area? Well, it's only got face categories at its top end. So to ask that question, what we do is put a bunch of different objects into here, run it through the system, and ask whether the features in the penultimate layer can be used to, with a linear SVM to decode those object categories. And when we do that, we find that indeed this VGG face that's been trained only on face recognition can in fact decode object categories above chance. It's not great at it, more on that later, but it's above chance, much like the fusiform face area. So what that means is that above chance object recognition abilities doesn't mean that a system is not optimized all and only for face recognition. Ditto for the fusiform face area. So the real question then is the fusiform face area used in object recognition? So to answer that question, deep nets won't help us, and in fact, functional MRI won't help us. We need to use a causal test. Luckily, a few years ago, I got a phone call from a collaborator telling me that this man here who was then in a hospital in Japan with electrodes all over the surface of the bottom of his brain, shown here. Here's his brain, here's the location of electrodes. And for comparison, here's the bottom of my brain, showing in red my fusiform face area, in green my parahippocampal place area, and a little purple region that responds preferentially to color. This man had electrodes in his brain because he had severe epilepsy, and the neurosurgeons needed to map out functions in these regions before conducting neurosurgery to try to treat the epilepsy. So they wanted to know if I wanted to collaborate. Well, duh, of course I wanted to collaborate. So everybody in my lab dropped what they were doing, and we constructed some stimuli and shipped them to Japan so that we could then get the responses to each of these electrodes in here to a bunch of different classes of stimuli. We sent them faces and objects, but also Japanese characters, line drawings of objects, digit strings, pictures of bodies, etc. And here's the response of those electrodes along the fusiform gyrus to each of those categories of stimuli. So for example, these electrodes right here, in a position that looks quite comparable to my fusiform face area, here's their response over time to each of the categories. In red is the response. Again, now we're recording electrically right from the surface of the brain. This is much better than functional MRI. So now we can see time. This is 2 seconds across here, and this is the response to faces and the response to a whole bunch of different conditions. So if previously when I showed you the functional MRI responses that I was claiming were face specific, and you were underwhelmed, that's because functional MRI is kind of a crappy, blurry tool based on blood flow. When you have the real deal recording right from the surface of the brain, these sites are extremely selective for faces only. Okay, so that's just replicating and showing better data, better evidence for face selectivity. But what about this critical question of the causal role of these regions? Well, to find out, what neurosurgeons sometimes do to map functions is electrically stimulate different regions of the brain. And in this case, what we asked the patient to do was just tell us, excuse me, what happened and if anything changed when the neurosurgeons electrically stimulated those locations of the brain. So I'm going to show you a movie of what happens with this patient here. And I'm trusting Dobie to wave his arms if the movie doesn't work very well.
See, there's no sound. Is there supposed to be sound?
You can see in the lower right where the stimulation starts, right here. Recall this patient has no idea there's a fusiform face area. He doesn't know where this electrode is. Okay, that shows the causal role of that region in face perception. What we wanted to know is it causally involved in perception of other things. This is a kanji character for shortened here on a card. Okay, you get the idea. Could you see that more or less? Yes? Okay? Okay, great. Good. So the point is that this region not only responds very selectively to faces, but its causal role is extremely specific. Even when the patient is looking at an object that isn't a face, it doesn't change the shape of the box or the ball or the kanji. It just puts a face on top of it. Now, you might think, um, we're social primates. We're primed to see faces. Maybe anything you do to the brain, maybe you hit the head up here, you see a face. So how specific is this? Well, we can ask that by asking, what if he's stimulated right next door in that color preferring region? So let me show you what happens when he's stimulated there. Again, recall here he's getting stimulated. He has no idea there's a color preferring region or what electrode is being stimulated. And yet he says, okay, so really these things do just one thing? Yes, really. Some of them at least just do one thing, as you could see in that video. At least I think that's pretty clear now for the fusiform face area. Even though there's non-preferred information in that region, as Haxby showed and I and others have replicated, that's what we expect from even the most specialized face processor. We saw that with the VGG face trained network. So that's not evidence against the face specificity of the region. And even more strongly, even if there's information there about non-faces, that information doesn't seem to be read out, as you can see from the causal test. So I want to go on with a second, very different question, and that is, how is all this systematic structure built up in the brain over development? I find it impossible to look at this diagram here and not wonder how you start with a bunch of genes and end up with this systematic structure that's so similar in every person. I'm not going to answer that question, but I'll dance around it a little bit and tell you about some cool data. So the first question you might think of is, okay, when does all that structure arise in development? And so many studies, starting from some pioneering work of Colony Spector way back in the over 10 years ago, studied kids aged 4 years and up. And they showed that a lot of the structure is already present, at least in the ventral pathway, by age 4, although it's still developing well into the teenage years. But that's late for development, right? And so then, after many years of effort, the remarkable Rebecca Sachs and her colleagues, this is Ben Dean, this is Hillary Richardson here, after many years of effort trying to figure out how to scan infants, including going to the great length of producing her own subject. This is little Arthur here. What Rebecca did was figure out how to get sensible data from infants. And she showed, for example, here's an infant brain showing differential responses to faces in orange and scenes in blue. And for comparison, here's an adult brain with the same contrast. And you can see that this 6-month-old infant here has a recognizably similar layout of cortical regions that respond preferentially to faces and scenes. Rebecca also showed that those regions are not as selective in infants as they are in adults. Their response profiles are quite different, but the general layout of the cortex is already recognizably adult-like for at least some of these regions by 6 months of age. So that's totally awesome. But really what we want to know is not just when these regions arise, but how. And that's a much harder question. So I want to tell you in a bit of detail about some work we've been doing recently, and by we, I mean my postdoc Ratan Murthy, tackling 2 basic questions about cortical development, focusing on face selectivity in the fusiform. First question is, how does selectivity for faces arise right there in development? And here the standard hypothesis for some time has been that face selectivity is learned from our visual experience with faces. And in fact, a very recent paper made that exact claim. The title of the paper was "Seeing Faces is Necessary for Face Domain Formation." That was an amazing study in monkeys looking at the face selective patches analogous to this one that have also been found in monkeys, and showing that those face selective patches do not arise in monkeys who are raised by human caregivers who have welding masks over their faces, such that those monkeys never see faces. They have lots of other visual experience, lots of auditory and other social experience, but they have never seen faces, and indeed they don't develop these fusiform face regions, hence leading to this apparently very reasonable conclusion that seeing faces is necessary for face domain formation. However, about the same time that this paper was published, another paper was published with the nearly opposite conclusion in its title. Vandenherk et al. Published a paper saying development of visual category selectivity in ventral visual cortex does not require visual experience. And that was based on a study of congenitally blind people listening to different categories of visual stimuli and finding weak but significant differential responses to face-related sounds and scene-related sounds and body-related sounds. So this question is still in play. Now, the second basic question we addressed here was, why does face selectivity land so systematically in that location right there? The standard story for the last bunch of years has been that face selectivity, this is a good patch of cortex for face selectivity, because when we look at faces, we foveate them. We look straight at the face on our fovea. That's what we tend to do, and indeed we do that from an early age. And indeed, it's known that foveal retinotopic cortex is approximately back here, and there's preferential connectivity, even at birth, from foveal retinotopic cortex right up there, giving a pipeline for faces to come straight from the fovea right up to that patch of cortex. In addition, it has been argued, with some evidence, that this patch of cortex seems to have a preferential response to curvy shapes, not just faces, but as a weak response to other curvy features. And so maybe that curvy preference is present at birth and also serves to preferentially produce a response to faces here from which experience could take it from there. Or at least these two hypotheses were the kind of standard and I thought very sensible hypotheses until the Vandenherk paper came along, and some other data that doesn't quite fit with this. So we figured this was worth another crack. And so our approach was to test congenitally blind people, but use haptic stimuli. We thought haptic stimuli, because they have shape, you can perceive a shape both from vision and from touch. We thought that might be an interesting way to go at this question. So what Ratan did was to generate a set of 3D printed stimuli about yay big, they're a couple inches high, just like these shown here. And these stimuli were several different faces, mazes, hands, and chairs. We then popped subjects in the scanner, and with this magnificently high-tech device composed of a disc of cardboard poked onto a CD holder, I think the materials cost 99 cents for this device, with a postdoc outside the scanner bore. If you guys haven't scanned, you might not recognize, this is a subject lying in the scanner bore here with this thing lying on their belly. And there's a postdoc situated just outside the bore rotating that stimulus every few seconds so that the subject's hand can be placed there and the stimuli rotate by, giving the subject a chance to palpate each stimulus one at a time as it goes by. Okay, so that's the design. Then control-sighted subjects also perceived the same stimuli, but they saw them visually, much like these little images of these same stimuli rotating in depth like that. So that's our basic design. And so the first question is, can we see nice face selectivity in the sighted subjects when they see the stimuli? And indeed, here are two individual subjects showing the bottom surface of their brain. This is the fusiform face area, and these two subjects, they're shown in yellow. And if you look at aligning the data across subjects to see where that face selectivity most typically lands in our group of 15 sighted subjects, you see that it lands in the canonical location right there in the fusiform gyrus. And you also see something that was pointed out a few years ago by Kevin Wiener and Colony Gross-Spector, that there's a teeny little sulcus called the mid-fusiform sulcus that runs down the middle of the fusiform gyrus, and the face selective activations land systematically lateral to that sulcus. That's a nice anatomical landmark that we can use. So now the key question, what about congenitally blind subjects feeling these stimuli? Well, here are two blind subjects, and here is their preferential responses to feeling faces versus feeling chairs, hands, and scenes. And you can see that their face selective activation looks not all that different from the sighted subjects. And when you pull the data over, we saw this in most but not all of our blind subjects. When you pull the data across the blind subjects, you see that that face selective activation also lands in a very similar location, systematically lateral to this anatomical landmark of the mid-fusiform sulcus. Now, if we take those data and subdivide them in two, use half the data to identify the region and left out data to quantify its selectivity, you see the standard face selective response in sighted subjects, seeing the stimuli. What about the blind subjects? Well, it's remarkably similar. The face selective response is very similar in selectivity, although notice the absolute magnitude of the response. I don't know if you can see this, but it's a 1% signal change for faces for sighted subjects looking at the stimuli, and a 0.4% signal change for blind subjects feeling them, and that's an important difference. But the selectivity is very similar in the two groups. So apparently, seeing faces is not necessary for the development of face selectivity in the fusiform gyrus. So, so much for this very sensible hypothesis. It just isn't true empirically. Now, does this mean that the face area is innate? No, not necessarily. Blind subjects do have experience with faces. They feel faces. So we can't say that there's no relevant experience. However, that said, blind subjects don't feel faces a lot. When I was first investigating this, I Googled this question. I found all these websites put up by blind people saying things like, how come sighted people are always asking if we want to feel their face? That's disgusting. So it's not a thing. It's not done much. But blind infants surely feel their mother's faces. Some of them have dolls. They feel their own face. There's some experience feeling faces. And so we can't use this to argue for innateness fully. But they certainly have no visual experience with faces and no visual expertise with faces. What about this second hypothesis, that it lands in that location because that location has a foveal bias and hence the stimuli get piped up there? Well, that doesn't make any sense because our congenitally blind subjects have no foveal experience. So that can't account for why it lands in the same location in the blind subjects. What about this idea of this curvature bias, this kind of shape map? Well, maybe that shape map is amodal. Maybe it could respond to curvy shapes. So we tested that next by bringing back a subset of our congenitally blind subjects and giving them the same experiment, but adding in spheroids, curvy shapes, and cuboids, rectilinear shapes. So the first thing we did is we localized their face region from session 1 to see if we could replicate their face selectivity in session 2, and that worked very nicely here. I love replicating results, so that makes me very happy. But of course, the key question here is there a higher response to these spheroids and cuboids, as you might expect, if that region has a curvature bias? And there isn't, if anything, the opposite. So no evidence here for curvature bias. Another question, if this region is about amodal shape in some abstract sense, then it shouldn't respond to auditory sounds, which don't directly reveal shape in the way that vision and touch do. So we asked whether we could replicate that kind of wild result from Vandenherk. Frankly, it was done by a really excellent lab, so I had to trust it, but the result was so astonishing it was hard to believe. So we got their same stimuli and asked if we could replicate their results. And we got a spectacular replication of their results, showing a much higher response in that exact same region when subjects hear face-related sounds compared to sounds face-related sounds were not verbal sounds. They were things like sneezing and chewing and kissing, compared to sounds like, waves and traffic and wind and trees and stuff like that, and body sounds and object sounds. So all of this shows we can replicate the Van den Herk result, and these data are really hard to account for by a general preference for curved shapes in that region. So we've ruled out all the standard hypotheses, all the sensible hypotheses. So now what? So now we've turned to another hypothesis based on connectivity, long-range connectivity of the cortex. And this is actually a long-standing idea in the field that different regions of the cortex have connectivity fingerprints. That is, each functionally distinct region has a specific set of connections to distant other regions of the cortex. So, for example, some little patch of cortex might have strong responses to, say, those distant regions of the cortex, but not others. And this is an idea that's been kicking around in the field for a very long time. But most recently, my then postdoc, Zeynep Sagan, decided to use diffusion tractography that gives us a highly imperfect, but better than nothing, measure of long-range connectivity in the brain. So she used these connectivity fingerprints. Actually, she started this work before she joined my lab, so I shouldn't be claiming credit for her. This is her work. She showed that those connectivity fingerprints can predict the precise location of face selectivity in the same subject. And then she and others showed since then that it can predict the locations of many functionally distinct regions. So the hypothesis that I want to consider here is that these long-range connectivity fingerprints play a causal role in shaping the functional development of these regions over infancy and childhood. And in fact, there's already evidence for this idea. The long-range connectivity of each region seems to be, to some degree, present at birth from histological studies post-mortem. And Zeynep Sagan and I also showed that you can predict the location of a functional region that arises much later. So we showed that visual word form area I mentioned at the beginning of the talk, it only shows up after kids learn to read. So we found it in kids age 8. You could identify it clearly after they learned to read. But we had scanned the same kids when they were 5, before they learned to read, and we had collected diffusion imaging data at age 5. And what we showed is you could predict the exact location of that region at age 8 from their connectivity fingerprint of that specific subject at age 5 before they learned to read, suggesting that the precise connectivity of that region predicted and perhaps played a causal role in why the visual word form area landed in that exact location. In fact, other evidence for a causal role comes from classic work by Murgankar, who used what he called rewired ferrets. He surgically altered the anatomical connections in developing ferrets before their eyes were opened, with the result that he ended up piping visual information into what would have been auditory cortex. And the astonishing result is you found many of the functional features of primary visual cortex in what would have been primary auditory cortex when visual information was piped into that location. That's a really beautiful causal test of this idea. So we then asked whether these connectivity fingerprints might explain the similar locus of the face selective patch in the fusiform gyrus in blind and sighted subjects. And so we used So we asked whether that connectivity fingerprint was similar in blind and sighted subjects. And as a minor detail for the imagers, in this case, we used resting functional correlations rather than diffusion tractography to get a correlation fingerprint rather than a connectivity fingerprint. The details don't really matter. And so what we found was a specific set of connections in one set of subjects that predicts where face selectivity will arise, and then you test it in held-out subjects. So, for example, here is the observed face selective patch. The white outline shows the fusiform gyrus in this one subject. The color shows you the face selective regions using touch in one of our blind subjects, faces versus the other 3 conditions. Here, you see the predicted location of face selectivity in that subject using that subject's resting functional connectivity fingerprints trained with a prediction from sighted subject connectivity fingerprints. And so what you see is a very similar pattern. This face selective patch here is predicted from the connectivity fingerprint of sighted subjects applied to this subject's connectivity fingerprint. And that prediction works even better than taking, this is now a group analysis of the blind subjects, saying where in general is face selectivity in the blind subjects. And you can see that even though that's more similar to this blind subject, it doesn't predict it as well as the connectivity based on the fingerprints of the sighted subjects. And this just shows that this is true in all but one of the blind subjects. So if I lost you there, the bottom line is that actually we can predict the location of the face selectivity in blind subjects from this diagnostic connectivity fingerprint of the fusiform face area in sighted subjects and vice versa. So now the conjecture is that it's not just connectivity in general, but that it must be probably top-down connections from frontal and parietal regions that seem most likely to be instructing the location of the face area in the blind subjects. And there's lots of open questions. I'm sort of running short on time, so I'm going to skip over these. There are lots of questions about this that we haven't begun to answer. We could get back to that in the question period if you like. So I started with this question of how do you build all this systematic structure in development? I said that face preferences and their early arise in their characteristic locations very early in development, that they can develop there without visual experience, and that locus may be specified at birth by the long-range connectivity of each region of the cortex, yet to be shown with stronger evidence, but that's a conjecture. Now in the final section, I want to turn from this question of how the cortex develops to why does it have the particular organization it does in the first place? Why, from a computational point of view, does modular design make sense? And why do we have the particular specializations we do in the cortex? And so for many years, this is work with Katerina Dobbs, for many years I would end each talk with these questions and I would say, well, presumably we have specialized cortical regions for mental functions that can't be well done with generic processing machinery. So you need special computational machinery for those functions. But that was just kind of loose BS on my part. No, I didn't see any way to test it, and I didn't really anticipate. I had no idea how it would ever be tested. But then Katharina and I started thinking about this, and we thought, hey, this is also work with Alex Kell. We thought, why have special machinery for face recognition? Let's tackle this using convolutional neural networks. In particular, we wanted to test the hypothesis that the ideal feature space for face and object recognition are so distinct that one system can't do both without a cost. Now recall that I mentioned the beginning of this work at the beginning of the talk. At that point, my point was even a dedicated face system that's only been trained on faces, if you put objects into it, you can do above chance discriminating one object category for another. But now I want to make the opposite point, that actually object classification was pretty lousy in a dedicated face processor, suggesting that the features that are optimized for face recognition aren't that great for object classification. And we can also ask the opposite question. What about a network trained only on object recognition that's never seen faces? So Katerina took AlexNet again and trained it on a subset of ImageNet that had just objects and no faces and no animals. And she trained it on object categorization. And she found, and then she put faces into the system and measured classification performance, reading a linear SVM out of the penultimate layer. And she found the opposite. So a dedicated system trained only on faces is pretty lousy at object recognition, and a system optimized for object recognition is pretty lousy at face recognition. And that's consistent with our hypothesis that an optimized feature space for faces doesn't work that well on object recognition and vice versa. However, and maybe that's why we need different processors for faces and objects in the brain. However, this is not yet the strongest test. What if we tried to train one network on both maybe then we would find a common feature space that would work fine for both face and object recognition, even if it wasn't found by these networks trained on only one. So we decided to test that by testing a fully shared dual task network, by just putting all the categories at the end, all 1,000 faces and all 423 object categories. And we would train it on a batch of faces, then a batch of objects, then a batch of faces, then a batch of objects, and do this until it reached plateau performance. And now, since all of these networks here have classifiers at the end, we can now just look at accuracy and ask whether this dual task system performs worse on each task than the single systems. In other words, is there a cost of sharing both tasks in one network? So first, let me remind you or show you what the performance is on the single networks. So this is performance on face recognition in the face-only network and on object categorization in the object-only network. And you see first that in some sense, face recognition is actually an easier task than object recognition, which is an interesting result in itself. For years, the hypothesis about why we have dedicated face processing systems was that face recognition was just harder than object recognition, so you need extra tissue to do that extra task. Well, apparently not. But second, if you're worrying that our object classification performance is not that great, this is just AlexNet, and we're training it on far fewer categories because we're trying to be careful about the training diet here that has no faces in it, although and no animals in it. Okay, so this is decent performance given that. But the key question now is how does the fully shared network fare compared to performance on the singly trained network? And what you see is a huge cost of sharing. Face recognition performance in the shared network is much worse than face recognition performance in the face-only network. And same deal for object categorization. It also goes way down. So what we're seeing here is a big cost of sharing when you try to train one network at both. Consistent with our hypothesis that the ideal feature spaces for face and object recognition are different, so one system can't do both tasks without a cost. And so I think that suggests that may be why we have specialized machinery in our brain for face recognition. Now, of course, there's a whole bunch of questions to ask here. For example, can you share some of the stages without a cost? After all, that's what you see in the human visual system. You use the same V1 for face and object recognition, probably the same parts of V3 and V4. What about networks? Well, we can train a whole family of branch networks that share the first 1 or 2 or 3 stages and branch after that and ask how they perform. And there are lots and lots of other questions we want to ask. So just to summarize the conclusions here, we think face and object recognition may be distinct in the brain because there's no common feature space that can do both without a cost. Next, if this general line of work is on the right track, we have to predict a big cost of sharing for places and objects, because that's what we see in the brain, but not for, say, food and objects, because we don't see special regions of the brain for food recognition, and yet food recognition is an important thing that we do. In a prior line of work that inspired this work, Alex Kell and Josh McDermott showed that there's, using these same methods that we had adopted in our work, that there is a cost of sharing for speech recognition and musical genre classification. And maybe in the long run, we can ask whether all of the brain organization we've discovered with functional MRI might be explained in this way. Maybe once we figure out how to test it, we'll find that social cognition and language require distinct tissue that show a cost of sharing if you try to train them into the same network. I have no idea how to do that at the moment, but maybe in a few years we'll be able to figure it out, and many related questions. So I want to end this by re-acknowledging the 2 people who did the main lines of work I talked about here, Ratan Murthy and Katarina Dobbs, as well as my wonderful lab, who are very smart and have all kinds of skills that I don't, and most importantly, who are game to try all kinds of crazy stuff with me. And I want to thank you for your attention, and I'll stop there and take questions.
So if you have questions, Please just come to the front and look at that camera so that Nancy can actually see your face as you were asking the question.
Hi, my name is James. Thank you for your time. I will stand over here.
Thank you.
I have a quick question on the resources that you're using to recognize face and recognize objects and combine. So from a high-level perspective, if you have a particular type of machinery to do one and particular type of machine to do another, if you use the same type of resource to do both, like their effectiveness reduced by half. So is the third machinery's like neural net count, connectivity doubled, or the same compared to the first two? Because if you do any, like not face, not object, my hypothesis is it will be reduced.
Great. Okay, it's a great question. What we wanted to do was match the situation that you see with the single trained networks. So we trained on the same number of stimuli, and yes, those it is half the units on each side of the branch, right? And so the idea is if you can do that task with a shared network, you should be able to divide. You should be able to divide it into those halves. Now, it's an important question. At some point, if you have a big enough network, you're going to be able to do any task in there. One would think that with enough training and a big enough network, you should be able to, at the very least, discover the two separate networks, just put them together in the same single network, if you see what I mean. And so we're now we're running experiments with larger networks than AlexNet using VGG and asking whether we get similar costs of sharing. We are predicting that we will not get the same cost of sharing, maybe some, but it will be much smaller. So in that case, the question that we'll be most interested in, and we sort of have a glimmer of that, we haven't nailed it, but it looks like that's what's happening. In that case, what we're going to look at is whether, even if there's no cost of sharing, is that because the larger network has discovered that same division of labor, such that if we look in that larger network, we'll discover a latent division in the units along that network, such that if we take out the ones that are more that have stronger face weights, if we lesion them, we'll selectively impair face recognition and vice versa. So that's all ongoing work. I can't tell you the answer yet, but I agree with you. The size of the network will ultimately matter, and that's how we're going about addressing it.
Okay, thank you.
Thank you. Other questions?
Hello, my name is Shankar. So a lot of social interactions involve faces. So how much of, when you talk about the
Sorry, can you speak just a little bit louder? It's slightly hard to hear.
So a lot of the social interaction involves faces. So how much of the fingerprints that you see in your connectivity map onto regions that are, like, in general, linked with social interactions?
Yeah, it's a great question. I skipped over the methods used in the connectivity fingerprints. And it's a great question. We really want to know what those exact connections are. My view, based on a really disappointing 2 years trying to answer exactly that kind of question, using what should be the best diffusion tractography methods anywhere. There's a connectome scanner at NIH that is optimized for diffusion imaging, and that should give us the best ability to do tractography. I was trying to answer exactly that kind of question. And I am very disappointed in tractography's ability to give us a clear answer to whether two particular cortical regions are or are not connected, or how strongly they are. Tractography has many known limits, and I was trying to take it very literally for that question. I think just the question you asked. I don't think it can answer that question well. The reason that I'm still using the connectivity fingerprints nonetheless is I think that even if any one connection is so problematic, you can't necessarily believe it from tractography or, from that matter, from resting functional correlations. I think given enough connections, there's enough information in that pattern to be able to pull out a signal and predict where the regions will land. It's sort of analogous to using multiple voxel pattern analysis to pull out a signal, even if you don't believe that any one voxel has that tiny difference that's being used in the multiple voxel pattern analysis, if you see what I mean. So it's a very disappointing answer. I think we don't know the answer to that question, and it's driving me crazy. So I hope that somebody will invent better methods so we can say what exactly are those connections that are driving the connectivity fingerprints. Oh, I can say one thing though. We did take our connectivity fingerprints and at least ask, I think I have a slide on this, at least ask whether those predictions are which lobe they're coming from, because we have many anatomical parcels in each lobe. So let me just see if I can find that slide. Yeah, okay. So in this bottom right here, are my slides still up?
Yes.
Yeah, okay.
Yes.
Okay. So what this shows us is an analysis where we broke down that prediction, cross-trained from sighted to blind subject. Oh, this is within, okay, sorry. This is within subjects here, showing you the prediction accuracy from our correlation fingerprints in sighted subjects from just occipital anatomical parcels, just temporal, just frontal, and just parietal. All 4 lobes give you predictivity in sighted subjects. In our blind subjects, it's only the frontal and parietal parcels that give you that information. And I think that makes perfect sense because after all, they're not getting any relevant input from the occipital lobe and their temporal input is different. And so I think that the connectivity that's directing function in the blind subjects is the top-down connections from frontal and parietal lobes. That's going quite a bit beyond my data, but that's, I think, yeah.
Thank you.
Yep.
Wonderful. So we are running out of time, so let's thank Nancy one more time. And if you're a student who's meeting with Nancy for lunch, make sure to call her on Skype. Yeah, thank you, Nancy.
All right, thank you. Bye-bye.